Bismillahirrahmanirrahim. Assalamu alaikum dear all students. Welcome to the histotechniques. Today we will discuss about the tissue fixation of the tissues which are being removed during any surgery or during any operation. So, in today's lecture the outlines are the introduction about fixation, that what is fixation, why it is important, and what is the function of fixation. The second outline of today's lecture is the purpose of fixation. What is the purpose of fixation, why fixation is important, and in the third we will discuss about the different changes that occurs during fixation inside a tissues, that what are the side effects, what are the bad effects or the good effects of the fixation during fixing a tissue. And we will also discuss about the fixation of the large tissues when a tissues having size larger than the normal tissues, and also we will discuss about certain special tissues, for example brain or small biopsies type tissues. So, we will also discuss about that sort of tissues, and in the last we will discuss about the decalcification of bone, how bone is decalcified, how the minerals they are being removed to soften the bone. So, these are the outlines of today's lecture. Coming to the first part of the lecture, that is the fixation. Fixation is basically a process, a phenomena in which the cells or the tissue, these are fixed in a chemical and as well as a physical state. As we know that when a tissue is removed from the body, so all the oxygen supply, the food supply, they are being discontinued and that is disconnected. So, when any tissue that is removed from the body, then their enzymes, their all processes are stopped. So, when their normal processes are stopped and their respiration, their energy generation is stopped, so after certain time the death process starts in the tissue. So, to stop that death process and to stop that decaying process, this fixation is important. The enzymes inside the cells, for example the lysozymes, the granzymes, so they start releasing, and when they are released, so these enzymes then they destroy the mechanism of the cell, the structure of the cell, the physical state of the cell. So, fixation is important to stop all these destroying process, to stop all these damaging process. So, for that purpose fixation is important, and this fixation, it prevents all the biochemical activities and all the proteolytic activities in the cells, because when the cell dies, so certain biochemical activities they starts to destroy the cell, to decay the cell and to damage the important proteinaceous and non-proteinaceous parts of the cells. So, to stop that proteolytic activities, to stop damaging the proteins, the fixation is important to protect the cells from these sort of activities. During fixation, the tissue or the cells, they are resisted to any sort of change, either that is morphological change or the decomposition, because when we treat these tissues with the fixation, with the fixatives, so after that fixation when another type of chemical substance or chemicals they are being poured on the tissue, or when the tissue is being come in front of the tissue, or they make contact with these sort of chemicals which are usually corrosive. So, then after fixation these tissue they resist to any type of morphological, biological, proteolytic change within that. And this fixation, this is the first step of any type of tissue processing, any type of histological tissue examination, or any type of cytological tissue examination in the histopathology or cytopathology. So, this is very important that the tissue should be fixed properly, and that fixation process should be efficient to continue all our processes efficiently, effectively, and at the end we get good results from this process, and to study the tissues very clearly and effectively. So, for that purpose fixation is very important, and this is the first step. And the basic purpose, the basic aim of fixation is to maintain the tissue nearest to its original state in the living system. Just like, for example, if a muscle that is being removed from the living body, so to maintain that muscle just like in the living body, we have to fix that muscle. Now, what are the purposes of fixation? Why we perform fixation? What is the importance of fixation? So, the first purpose of fixation is, just like we discussed in our previous slide, that to preserve the tissue nearest to its original living state, because to study the disease, to study the tissue, it is important that that tissue must be in that original state or must be near to its original state. So, the first purpose of fixation is the preservation of tissue that is very close, very near to that original living state. The second purpose of fixation is to prevent any sort of change in the tissue, either that is morphological change, either that is change in the shape, change in the size, or change in the any sort of cells. So, to stop that any kind of cells, to stop any kind of change in the cells in the tissue, so this fixation process is performed. The third purpose of fixation is to prevent autolysis. Autolysis is the process in which the cells destroys itself by activating, by releasing different types of enzymes. So, these enzymes which are released in the cells, these enzymes destroy their own cells. So, this process is called autolysis. So, when fixation is performed, so this fixation stops any sort of autolysis, any sort of proteins, any sort of enzymes released from the cells. So, this is autolysis is also important to maintain. This autolysis stoppage process is also important to maintain the cells nearest to its original living state. The fourth purpose of fixation is to make the tissue firm to hard, because usually the tissue that is soft, so we cannot cut that tissue easily, and we cannot perform any sort of techniques on that soft tissue. So, for that purpose we fixes that tissue so that the tissue becomes slightly firm. And also a hard tissue is not important, it is not suitable for us, and the hard tissue is also inappropriate to process. So, therefore, if the tissue is fixed more than the required time, then the tissue becomes harder. So, therefore, we need firm to hard tissue so that it can be processed adequately and we can get good results from the tissue. The next purpose of tissue fixation is to prevent any sort of bacterial or any sort of microbial growth in the tissue, because in certain tissues there is microbial flora. So, when these microbial flora they get proper environment, proper suitable temperature and pH and food, so they destroy the tissue by starting proteolytic activities and releasing their enzymes. So, these bacteria or these fungi they destroy the tissue after the tissue when it is removed from the living body. So, one purpose of fixation is prevention of the bacterial or fungal or microbial growth in the tissues. And one important purpose of tissue fixation is to have clear staining, because before microscopic examination the tissues are being stained with the hematoxylin and eosin staining. So, for that staining purpose the proper fixation is important. Therefore, the tissue is fixed before processing. And the last purpose of tissue fixation is to have better optical quality of the cells. When the tissue is being examined under microscope, so the clear picture of the cells it depends on the fixation. When the fixation is proper, when good fixative is used for proper time and it is then processed, that tissues become look very clearly and we can have a good diagnosis, we can study the tissue very clearly after fixing the tissue clearly. Now, what is ideal fixative? What are the properties that should be present in an ideal fixative? So, the first one is ideal fixative, it should prevent autolysis, as we have already discussed the autolysis. So, it is the property of an ideal fixative that it prevent autolysis for starting or prevention of any sort of destruction within the cells within the tissues. And the second property of an ideal fixative that it should prevent any sort of bacterial decomposition or the destruction of the cells, the tissue by bacteria or the microbes. So, ideal fixative must have this property to stop, to prevent bacterial or microbial decomposition. And the third property of an ideal fixative, that this fixative should maintain the volume, the shape, the size of the tissue, because if there is any sort of change in the volume or in the size or in the shape, so this tissue get destroyed and we cannot get good result by studying that type of tissue microscopically. So, the ideal fixative it should have this property to maintaining the volume, the shape, the size of the tissue. And ideal fixative should have the property that after fixing the tissue with that fixative, the staining should be good and it should, the tissue must get good stain so that it gives us a good result. So, if any tissue that is fixed, but the staining is not adequate, so this type of fixative should not be used. And another property, another quality of an ideal fixative, that the ideal fixative must have rapid action on the tissues, and all these processes, all these activities, all the actions they must be fast in their processing, because with the passage of time the tissue get shrink or the size, there is variation in the size, variation in the volume. So, the rapidly active fixative and the fixative which have fast action that prevent any sort of disturbance, any sort of decomposition, the autolysis. So, therefore, ideal fixative must have a rapid mode of action to maintain, to fix the tissue in a short time, and it should be cheap in price, because this is very important that the fixative have economic impact. So, any fixative which have all these properties and also cheap, so this is a good fixative. It should not be costly. So, cheap fixative is ideal, having all these properties. And the last property of ideal fixatives is that it should be not non-toxic to the tissue as well as to the person who is dealing with this fixative. So, these are the properties of an ideal fixative that should be used for the fixation of the tissue in the histopathology lab.